Hello. In this video, we'll be explaining the biotechnology technique of microarrays and how it has been used to better understand and possibly treat polycystic ovary syndrome, or PCOS. We'll explore the molecular mechanisms of microarrays, the kind of data they generate, and how they compare to other techniques. We will also go over an example of their use in PCOS research. Now, let's dive deeper into how microarrays work at the molecular level. This technology is used to measure gene expression across thousands of genes simultaneously. By comparing the gene activity in healthy individuals with those who have a condition like PCOS, we can identify which genes are behaving differently. The microarray process can be broken down into several steps. The first step is sample preparation. This is collecting biological samples. In the case of PCOS, this could be granulosa cells from the ovaries, as these cells are directly involved in the hormone production and follicle development. These samples are chosen because they hold the key to understanding how gene expression is altered in PCOS. The next step is to extract the RNA from the cells. RNA is critical because it reflects which genes are being actively transcribed into proteins at that moment. The genes producing more RNA are considered to be more active, and this gives us a snapshot of cellular activity. In the context of PCOS, we are interested in genes involved in hormone synthesis, such as CYP11A1 and CYP19, which regulate steroid production and could be over or underexpressed. RNA is not stable enough for experiments, so we convert it into complementary DNA, also known as cDNA, using the enzyme reverse transcriptase. This process copies the RNA sequence into a stable DNA version, which can then be studied. The cDNA acts as a long-term representation of gene expression, allowing us to study the gene activity without worrying about RNA degradation. To track the expression levels of different genes, we label the cDNA from each sample with fluorescent dyes. Typically, two colors are used, one color for the control sample from the healthy individuals, and one for the PCOS sample. For example, red dye might label the cDNA from PCOS patients, and green dye would label the healthy control group. When combined, this allows us to compare gene activity directly. The labeled cDNA is then applied to the microarray chip. Each spot on the chip contains a short DNA sequence called a probe, which corresponds to a specific gene. Think of the chip like a grid with each spot representing a different gene. The cDNA from the sample will bind or hybridize to the spots containing complementary sequences. The more cDNA that binds to a spot, the more active that gene is. After hybridization, the chip is washed to remove any unbound or loosely bound cDNA, ensuring that the signal is specific and accurate. A laser scanner then detects the fluorescent signal on the chip. If the gene is more active, you'll see more of the red fluorescence if it's the PCOS patient. If it's more active in the control, you'll see more green. If the gene's expression is unchanged, the two colors will mix.
Created using Powtoon.